Pancreatic cancer is one of our greatest challenges in cancer medicine. However, treatment standards are still based on conventional chemotherapies, like FOLFIRINOX, that deny individual tumor characteristics and only slightly improve survival times.
Particularly, pancreatic cancer is characterized by its inter- and intratumorous heterogeneity, caused by few driver mutations, such as oncogenic KRAS and a bundle of passenger mutations that define subtypes. The unstable subtype is supposed to be sensitive to a platinum therapy and frequently harbors mutations in DNA damage repair genes such as BRCA1/2 or ATM serine threonine kinase.
So the entire story of this gut paper started a couple of years ago when we got interested to work on a tyrosine kinase termed ATM, ataxia-telangiectasia mutate, which is one of the key players during DNA damage repair to repair DNA double-strand breaks. And we got access to a conditional knockout mouse which we crossed into the classical KRAS model driving pancreatic cancer formation. And what we found in the first paper that those tumors lacking ATM expression are more aggressive, they undergo EMT, epithelial mesenchymal transition, and when we translate this knowledge to humans, we also found that the overall survival in historic cohorts was lower when the tumors are lacking ATM. So in follow-up work, we found that when we treat those tumors lacking ATM, those mouse tumors lacking ATM with PARP inhibitors, there is obviously synthetic lethality and we can dramatically kill or stop tumor growth. However, in those first experiments, we noted that after prolonged treatment with the PARP inhibitor, the tumors can grow again. So obviously there is resistance or the efficacy of the treatment is not optimal yet. So we started and asked ourselves, how can we on the one hand overcome upcoming resistance and on the other hand improve therapy, and we started screening and came up with the triple therapy which we now tested in this paper.
Based on combinational synergy screening, we tried to endeavor a genotype-tailored targeted therapy in ATM-deficient pancreatic cancer. And indeed, we found synergy upon inhibition of PARP, ATR and DNA PK, resulting in synthetic lethality in murine and human pancreatic cancer. This we further refer to as PAT.
Mechanistically, PAT induces PARP trapping, replication fork stalling and mitosis defects, causing a p53-mediated apoptosis. Using human pancreatic cancer cells, we could ascribe homologous recombination defectiveness by chemical inhibition of ATM and thereby sensitize those cells to PAT with long-term tumor control in vivo. The efficacy of our PAT triple therapy combined with the inhibition of ATM was ultimately validated in a set of patient-derived xenografts and patient-derived organoids. We further exploited chemoresistance to single PARP inhibition and found that arising PARP inhibitor-resistant ATM-deficient cells were aneuploid and underwent epithelial to mesenchymal transition. Acquired multi-drug resistance came along with upregulation of drug transporters and a bypass within the DNA repair machinery. All these functional observations were mirrored in copy number variations affecting a specific region on chromosome 5, comprising several of the upregulated multi-drug resistance genes. Using these findings, we also proposed alternative strategies to overcome this chemoresistance, as inhibition of the DNA damage repair protein DNA ligase 1.
The presented preclinical approach paves the way to further personalize pancreatic cancer therapy by multi-drug targeting of the DNA repair machinery.